Today I'll be presenting my work on a microfluidic vascular graft on a chip platform, which enables testing of vascular graft materials in surfaces under physiological-like flow conditions. So in case you aren't aware, I'll provide a bit of a context. So vascular grafts are essential for managing cardiovascular disease in stenting and bypass applications. However, many of these devices ultimately fail due to thrombosis or clot formation on the blood contacting surface. And this has incentivized the development of anti-thrombogenic surfaces and materials for vascular applications. And one such technique which has shown promise in recent years has been the application of coatings. So in this investigation, I'll firstly describe the vascular graft on a chip platform and then outline a proof of concept investigation that we performed, which probes the thrombogenic performance of a surface enhanced coated graft as well as an unmodified control. So the vascular graft on a chip itself is fabricated using a very straightforward method, and it consists of a sandwiched assortment of materials with the middle layer serving as a thin film plastic adhesive from which our microfluidic channels are cut. Importantly, the microfluidic channels are cut to a specific geometry, which when subjected to flow, induce a physiologically relevant shear stress profile. And this is dependent on the geometry that we cut with a laser. We then apply our liquid-based chemistry to induce our anti-thrombogenic test surface in parallel with our unmodified control. We then test the thrombogenicity by perfusing whole blood, which has been spiked with fluorogenic markers and a dilutant agent, which enables real-time observation of channel occlusion over time. So this is demonstrated in the experimental section, where we firstly validate the fluidic regime using COMSOL. We obtained a mean shear stress of 1.5 Pascal, which is in agreement with the reported value for lower extremity bypass, which was the targeted application for this part of the project. We then probed the thrombogenic performance of our surfaces under flow over a series of time points. We tracked fibrin aggregation, shown in green, as well as thrombin activity, shown in blue. We also have the endpoints in the far panel F, which shows channel occlusion. This observation was then quantified in panel G, where we observed a statistically significant difference in thrombogenicity, which was in relation to post-perfusion fibrin adherence. This was further elucidated in panel H, where we did a post-perfusion surface analysis using SEM. And as you can observe, there's a marked difference in the fibrin density as well as the overall morphology of the clot structure on the surfaces. We then took our investigation a step further, exploiting the versatility of our fabrication method to test an additional fluidic regime with an additional biological species and an additional testing surface condition. So in this investigation, inspired by the branched aortic stent graft, we designed a channel geometry which incorporated variations in the channel diameter as well as curvature, which recreated a relevant shear stress profile. Then using our prior established methodology, we examined endothelialization as well as thrombogenic performance over time. And this is an ongoing investigation, but it really illustrates the versatility of our method and the vast potential for targeted applications. So in conclusion, we generated a novel vascular graft on a chip platform, which is generated using a simple and versatile 3-step protocol, generates physiologically relevant shear stress profiles, captured statistically significant difference in thrombogenic performance, and enabled translation of diverse flow conditions and biological species. And as I mentioned, this is an ongoing investigation, but in the future, I envision this serving as a valuable benchtop tool, which can inform materials optimization, ultimately improving clinical outcomes for cardiovascular disease sufferers. So thank you for listening and for the opportunity. If you have questions, I'd be happy to address them.
Thanks, Veronica.
Thank you.
Dr. Bat, do you have any questions?
Oh, yeah. So I have a question. So what's the, I mean, you showed that with the surface coating, there's the reduced thrombosis formation. So what is the potential mechanism for that? Do
Okay,
you have
so
any clues?
yeah, the surface modification that we tested was lubricant infused surface. And I actually think it was developed at WIS Institute. My supervisor is from WIS Institute originally. So the lubricant infused surface is a repellent surface. It's a biocompatible repellent surface. So that's the proposed mechanism of action. I definitely think it's worth investigating further in this dynamic regime to further understand the mechanism of action, but its previously described mechanism is a repellency. Yeah.
Thank you.
You're welcome. Thank you.
Just a quick question about the publication. Yeah. Have you tested the surface roughness before putting the functionalization of your channel?
So are you referring to the fabrication method or applying?
I think you're using laser, right? So with the laser, you do the fabrication.
Yes.
And then how do you consider the impact of the surface roughness of the channel?
So I guess roughness is a consideration in a number of ways. So in terms of the substrate itself, ePTFE, it did have roughness and porosity, but that wasn't a problem for the binding robustness because this is a blood repelling surface. In regards to the channel roughness, that was a concern because the laser does not have an entirely clean cut, but I did an ethanol sonication afterwards, as well as an inspection with the microscope to just quantify the extent of roughness on the cut edges. I don't know, did that answer your question?
Yeah, sure. And in terms of putting this into commercialization, so you mentioned that this should be a lab tool, and
well, in our lab, we do a lot of development of interfaces and biomaterial surfaces. So I envision in a lab such as ours that we can apply this to test our emerging materials in biological applications. So
okay.
that would be my vision anyway.
Thank you.
Thank you.
Dr. Bat.
Thank you, Veronica, for the great presentation and poster. I actually have two questions. One, and maybe it's in there and I missed it in your talk and in the poster, but what anticoagulant did you use for the blood when you run your system?
Okay, so this was donor whole blood that was citrated. So I recalcified the blood and it was perfused in a recalcified state.
Okay, great. The other question that I had that related to the last part of your experimental setup where you started using endothelialization, I believe, or endothelial cell. I was trying to understand, and maybe I missed it, what you were trying to do around this, just because we know with endothelialization, what we see in vitro is so far compared to what we see in vivo. And so I was trying to understand what your hypothesis was and what you're trying to test to understand what's the goal.
Yeah, so for that particular aspect of the experiment, we introduced a coating, which was an antibody. It had antibodies incorporated into it. And the goal with that coating was to promote endothelialization and suppress thrombogenicity. So we just broadened the scope of our performance testing to incorporate endothelialization as a parameter. So it was mainly to look at the performance of the coatings mostly, and whether or not.
Oh, sorry. So were you then expecting to capture like progenitor endothelial cells, or did you put endothelial cells? That's what I'm just trying to understand.
Oh no, we did a culture. We did a pre-culture with the endothelium. Observed the capacity of the surfaces to endothelize with different coatings, and then performed our clotting experiment. Afterwards. Yeah. Okay,
great.
Thank you very much.
All right, thank you, Veronica.
Thank you.